Hello and welcome to this video on the stages and conduct of a clinical trial. Clinical trials are the practical stage of product development. A drug lead or novel application has achieved an overwhelming body of evidence, whether published or otherwise. Here, the product is shown to work in healthy volunteers and in the target demographic. This consists of 5 primary stages. This is done to ensure safety, minimal impact, damages, and loss of life, whilst providing the greatest benefit should the trials go wrong. Before the trial even begins, there is preclinical investigation. This is testing of drugs in non-human subjects. It gathers efficacy, toxicity, and pharmacokinetic information. While preclinical research answers the basic questions about a drug's safety, it does not substitute for studies of ways the drugs will interact with the human body in a more complex situation. These extensive preclinical investigations involve both in vivo and in vitro studies of both animal and cells. The use of wide-ranging doses provide information and obtain preliminary direction as to how much and how to give medication that is being investigated. The first phase is also known as phase 0. This is an optional exploratory study, sometimes referred to as pilot studies. These come before phase 1 and are used to test how the body responds to an experimental drug. In these studies, small doses are given once or for a short time to a very small number of people. Phase 0 is a recent designation and was provided by the FDA's 2006 guidance. Phase 0 trials are also known as human microdosing studies. They are designed to speed up the development of a promising drug. It establishes very early on whether the drug or agent behaves in human subjects as was expected from preclinical studies. The biggest difference between phase 0 and later is that there is no benefit to the volunteer, as any benefit that will come from this study is many years later. This will be for other people because the drug doses are low. It should have no significant impact upon the person receiving it at this stage. This microdosing regime is more or less the basic situation in which the patients from the 2 Atom Ruins Everything videos found themselves. The experiments and trials are built to specify the following: who qualifies to participate, how many people will be part of the initial study, how long the study will last, whether there will be a control group and other ways to limit the research bias, how the drug will be given and at what doses, what assessments will be conducted, when and what data will be collected, and how the data will be reviewed and analyzed. The FDA or similar agencies provide approval for the trial based on the following: animal study data and toxicity, particularly side effects that cause great harm, the data, manufacturing information, clinical protocols, data from any prior human research, and information about the prior investigation. The application is then reviewed by a panel of experts including a project manager, a medical officer, statistician, pharmacologist, pharmacokineticist, chemists, and microbiologists. The first phase of actual clinical research is phase 1. Phase 1 clinical trials are done to test a new biomedical intervention and is for the first time in a small group of people, between 20 and 80, and this evaluates safety, particularly determining a safe dose range and significant side effects. Phase 1 trials were formerly referred to as first-in-man studies, and in the 1990s, gender neutrality is brought in with first-in-human studies replacing this. These trials are the first stage of testing in human subjects, determining the maximum amount of the drug that can be given. And this is particularly relevant if you are trying to get the most effective treatment with the highest possible dose before adverse effects make it untenable. The single ascending dose is also known as phase 1A, and this is the first part where a single dose is given to a person, and it is at a very insignificant level, and then that is given to a second person, and roughly 3 people are given this dose at this stage. Then 24 hours later, another group of people are given a slightly larger dose, until they begin to exhibit side effects, or the expected required dose and expected maximum possible dosing amount are reached. At this point in time, it is considered acceptable if there are no adverse reactions, or if the adverse reactions are minor. Then it reaches the stage phase 1b. 1b is multiple ascending doses, where clinical trial participants are given a single dose, and then a short time later they're given a second dose, both of these being small. These low doses continue in volume and number over a period of time until, again, the desired and expected pharmacokinetic requirements are met. And then, if there are no significant adverse reactions, it is considered a success, and it moves on to the next stage. Phase 1 trials normally last several months. They often have the first few people in the study getting a very low dose of treatment, and these people are watched very closely. And if only minor side effects are found, then the phase 1 is considered a success. Phase 1 focuses on looking at what the drug does to the body and what the body does to the drug, as some drugs, once they are metabolized by the liver, can become quite toxic, or some drugs can't be handled by the body and therefore the liver removes them before they have time to take effect. Safety is the main concern at this point, with doctors keeping a close eye on people and watching for anything serious. This may also be where rare side effects are not identified because there are not enough participants. Normally placebos are not used at this stage, and these studies usually include a small number of people. These studies are usually done in major research centers, and they are not designed to find out if the new treatment works against a cancer, for instance, but rather to see whether or not it has any effect on the individual who is healthy. Phase 2 trials are the next stage. Phase 2 clinical trials are done to study an intervention in a larger group of people, normally several hundred, to determine its efficacy, and to further evaluate safety. Once a dose or range of doses is determined, the goal is to evaluate whether the drug has any biological activity or effect. In theory, this has already been demonstrated in an animal model. Phase 2 trials, performed on larger groups, are designed to assess how well the drug works, and to identify any particular safety net requirements that may have been missed in the small number of Phase 1 participants. Genetic testing is becoming common, particularly when there is evidence of variation in metabolic rate, and when the development process for a new drug fails. This normally occurs during Phase 2. This is often owing to having toxic side effects. Some Phase 2 studies are divided into a Phase 2A and 2B, although there is no actual definition for how these two categories are divided. It's normally accepted that Phase 2A studies are pilot studies, demonstrating clinical efficacy or biological activity, being proof of concept. Phase 2 look to find the optimum dose at which the drug will show biological activity with minimal side effects. In the first stage, the investigator attempts to rule out drugs which have no or little activity. This stage of the investigation can last from several months to 2 or more years. The primary idea behind it is to get efficacy and side effects. Up to 33% of drugs move to the next phase from here. Usually, a group of 25 to 100 patients with the same disorder get the new treatment in a Phase 2 study. They are treated using the dose and method found to be the safest and most effective in Phase 1 studies. In a Phase 2 clinical trial, all the volunteers usually get the same dose, but some randomly assign participants to different treatment groups. These groups may get different doses or get the treatment in different ways, and this is often done to remove researcher bias. No placebo or sham group is used at this stage. Phase 2 studies are often done at major research centers, but may also be done in community hospitals or even doctors' offices if they are well connected enough, if the trial is wide enough. The next stage are Phase 3 studies, or Phase 3 clinical trials, and these are done to find the efficacy of an intervention in large groups of trial participants. These can range from several hundred to several thousand, and it compares the intervention to other standard or experimental interventions, and in some cases where there's a control group, there is a non-interventional care provided. Phase 3 studies are also used to monitor the adverse effects and to collect information that will allow the intervention to be used safely and possibly with greater effect. This phase is designed to assess effectiveness, and thereby its value in clinical practice. They are often randomized, with controlled multi-center trials being done on large groups of patients. This ensures that researcher bias and clinician bias is removed, and it depends very much upon the disease or medical condition that is being studied. There is a definitive assessment of how the effective the drug is, and it is compared with the current gold standard for treatment. Because of the size and relatively long duration, phase 3 trials are the most expensive and time-consuming. These are difficult trials to design and run, especially where therapeutical care is concerned, because there can be variation between standards and provision, especially where there is a chronic condition involved. Phase 3 trials of chronic conditions or diseases often have a short follow-up period for evaluation. This is particularly important if practice treatment requires many years of follow-up, and is sometimes called the pre-marketing phase, because it actually measures how well the drug is responded to by the public in general. These can go from 1 to 4 years, and they primarily measure the efficacy and monitoring of adverse reactions in a large cohort. Of the drugs that are developed at this point in time, between 25 and 30% move to the next phase. Examples of trials like this are the HTN-1. These studies are often done in many places across the country, or in the case of the HTN trials, across Europe. Phase 3 clinical trials are more likely to be offered by community-based oncologists, for instance, because they have access to a larger number of people to involve with these trials. These studies tend to last longer, and the placebo may be used in some phase 3 studies, but it's never used alone if there is an alternative treatment available that is known to work, simply because they can't provide one group of people with treatment and not the other, due to a whole range of ethical complications. Phase 4 is the last generally accepted phase of clinical trials. Phase 4 trials are generally done after an intervention has been marketed, and these studies are designed to monitor the effectiveness of an improved intervention after it's been given the green light by the FDA or a similar authority. They collect population data and information about any adverse events. These can be associated with widespread use of the drug from a long period of time. They may also be used to investigate potential use of it in different conditions, in combination with other therapies, or ways to improve it. A phase 4 trial is also known as a post-marketing surveillance trial, or sometimes as a confirmatory trial. Phase 4 trials involve safety surveillance, also known as pharmacovigilance, and ongoing technical support of a drug. Accelerated approval programs particularly require this, as phase 4 studies may be required by regulatory authorities, or may be undertaken by the sponsoring company for competitive reasons, for example finding a new market for a drug, like there is with Viagra, or for other reasons, such as the drug may not have been tested for interaction with other circumstances like pregnant women. This is the case in thalidomide. On certain population groups, such as pregnant women who are unlikely to subject themselves to trials, it's impossible to know before it occurs. Phase 4 studies look at drugs that have already been approved by the FDA and are available for doctors to prescribe for patients, but they might still be needed to answer important questions. Because it has involved thousands of people, small events that are significant may have gone without notice. It is typically the safest type of clinical trial, and the phase 4 studies look at safety over time, not in the short term as was the case with phase 3. These studies may also look at other aspects of treatment, such as quality of life or cost effectiveness of the treatment, and means of improving it and remarketing it, normally at a marked up price. So far there have been 5 stages of drug development, and it has only just got into market and is being observed for adverse events. As of about 2010, 50% of drug candidates either failed during phase 3 trials or are rejected by regulatory agencies. The amount of money spent on these phase 2 and phase 3 trials is significant, and depending on a number of factors and the particular area being studied, a clinical procedure can drive the prices as high as $20 million for phase 2 and as much as $53 million for phase 3 trials. The entire process to take a drug from initial preclinical development to the market can take 12 to 18 years and cost well over $1 billion. Much of this funding in the initial stages can come from federal governments such as National Institute of Health, Department of Defense, Department of Veterans Affairs, or private industry such as pharmaceutical and biotech companies. Medical institutions and foundations also often give money during the clinical stages, as it is their particular demographic, funding groups, or clientele that benefit directly from the funding for this particular form of research. As a consequence though, the research being conducted in the facility from a particular funding body tends to be biased, and there are further safeguards that need to be implemented in order to ensure that any reported findings have not been obfuscated or doctored to ensure further funding. As an example of this, there is Andrew Wakefield, who received funding from a particular group looking at his research, and then produced findings to match the expectations of that particular group. As a result, his work is well and truly discredited, more so for the damage it did after the fact. Thank you for watching this video. Please post any comments, questions, or suggestions you may have below.